Dear students, myself is Dr. Udham Singh Tiwana. I am working as Professor of Agronomy, School of Agricultural Sciences and Technology, RIMT University, Mandi, Gobindgarh. So, today we will discuss field plot techniques. Field plot technique deals with the various elements which are essential for a properly planned agricultural field experiment, such as size, shape, number of replications, experimental units, designs, and finally the statistical analysis of the data. So, now what is experiment? Experiment is the study of new facts or to confirm or deny the results of the previous investigation. Experimental design is the branch of statistics that deals with the design and analysis of the experiment. So, there are three basic principles of experimentation. Number one, randomization. Number two, replication. Number three is local control, also called as a blocking. The experimental designs are of two types: simple designs and complex designs. In case of simple designs, they are completely randomized design, CRD, randomized complete block design, RBD, Latin scale design, LSD. And the complex designs are factorial randomized block design, split plot design and strip plot design. So, we will discuss these designs one by one. So, number one is the completely randomized design. This design is applicable only when the experimental material is homogeneous, but our soil conditions in the field, they are usually heterogeneous. So, therefore, it is not a preferable method in the field experiments. It is generally applicable to the lab experimental conditions, where the environmental conditions can be easily controlled. So, second is the randomized complete block design. It is also called as RBD. RBD is used when the fertility gradient in the field is in one side. Therefore, it is the most commonly used experimental design in the agriculture. The local control is adopted and the experimental material is grouped into homogeneous subgroups called as a blocks. A block contains the entire set of treatments, and thus a block is equivalent to a replication. Blocks are made against the fertility gradient. Researchers have to follow all the principles of experimental design, such as replication, randomization and local control. So, it is the layout plan of the RBD. Suppose we have 5 nitrogen levels and 3 replications or the blocks. So, in the first set, the fertility gradient is in the vertical direction. So, we have made the blocks or the replications in the horizontal direction, whereas in the second set the fertility gradient is vertical. So, we have made it horizontal. So, we have made it vertical, the blocks or the replications. So, it is the analysis of variance table of RBD. So, source is replications. So, degree of freedom is R minus 1. So, in our first case, treatments are 5, replications are 3. In this case, R is 3 replications, 3 minus 1, 2, treatments T minus 1. So, degree of freedom for the treatments will be 5 minus 1 is equal to 4. In case of error, error is calculated as R minus 1 into T minus 1, which is equal to 8. So, total is replications into treatments minus 1 is equal to 14. 15 into 3, 15, 5 into 3, 15 minus 1, 14. So, in this case you see the error degree of freedom is less than 10, it should be minimum 10. So, to increase this error degree of freedom, there is provision in this design that either we can increase the number of replications or we can increase the number of treatments, so that the error degree of freedom is more than 12. So, in the second case, treatments are the same 5, but we have increased the number of replications from 3 to the 4. So, by increasing the number of replications, so the degree of freedom for the error, it becomes 12, which is sufficient for our experiment. So, in the third case, we have the same number of replications are 3, but treatments are 7. So, by increasing the treatments in the next case, so we also got the error degree of freedom is equal to 12. The factorial randomized block design, it is FRBD. So, this design is used when the factors are 2 or the more. In the previous case RBD, factor was only 1, suppose nitrogen. Here suppose you have 2 levels of the designs factors. So, number 1 factor is nitrogen, another is the phosphorus. So, in this case we will use the factorial randomized block design. So, the layout is the same as in case of RBD, but the statistical analysis is different than the RBD. Here in this case CD value is calculated for all the factors as well as the interaction, but CD value will be different for all the factors. It is the layout plan of the factorial randomized block design. Here nitrogen levels are 4. In the previous case, nitrogen levels was only 5. So, there were no other factor. Here there are 2 factors. Another factor is the phosphorus level. Phosphorus level are 3. So, in this case we will make the total treatment combinations. So, treatment combination will be 4 nitrogen levels into 3 phosphorus level. It is equal to 12. 4 into 3 is equal to 12. So, we have made the 12 combinations of our treatment. So, and they have been assigned to the each replication or the blocks. So, the analysis of variance table for the factorial randomized block design. So, here in this case replications are 3. So, it means the degree of freedom is 3 minus 1 2. In case of treatment A is 5. So, treatment A minus 1 is equal to 4. Treatment B it is 3 nitrogen levels. So, degree of freedom will be 2. So, then here we can also work out the interaction. So, treatment A minus 1 into treatment B minus 1. So, 4 into 2 is equal to 8. So, in case of error we will calculate by subtracting all the degree of freedoms of replication, treatment A, treatment B and the interaction from the total. So, total error degree of freedom will be calculated as by multiplying the treatments. So, 5 nitrogen levels into 3 phosphorus levels into 3 replications. So, these are the 60 minus 1. So, there are 60 minus 1 is equal to 59 will be the your total degree of freedom. The another design is the split plot design. This design is used when one factor requires larger plots than the others. For example, irrigations and tillage. So, each of the larger plot is split into the smaller plots to accommodate the other factor. So, in the split plot design, the larger plots are called the main plots and the smaller plots they are within the larger plot they are called as the subplots. The factor levels allotted to the main plots they are called as the main plot treatments and the factor levels allotted to the subplots are called as the subplot treatment. It is the layout plan of the split plot design. So, in this case the treatments are 4 irrigation levels and 3 nitrogen levels. So, irrigation it requires larger area. So, we have kept in the main blocks. So, here there are 4 main blocks. So, in the main each block the subplots has been allotted N1, N2 and N3 in all the main plots randomly. So, it is the analysis of variance table of the split plot design, 5 nitrogen levels, 3 phosphorus levels, total treatments are 5 into 3, 15, replications are 4. So, degree of freedom for the replication is 4 minus 1, 3. For treatment A, it is 5, 5 minus 1, 4. In treatment B, it is B minus 1. Here, we will work out two errors for treatment A as well as for the treatment B. For treatment A, error A, R minus 1 into treatment A minus 1, 4 into 3 is equal to 12. In case of treatment B, so here the error will be worked out by dividing the total error degree of freedom from all these replications, treatments, errors A, treatment B, interaction and error B also. The another design is the Latin scale design. This design is used when the fertility gradient is in the two directions. In this design, the experimental material is divided into rows and columns, and the treatments they are assigned randomly to the rows and the columns. The randomization is in such a way that each treatment occurs only once in each row and each column. So, suppose ignoring rows and considering the columns as a blocks, it gives an RBD design. Similarly, ignoring columns and treating rows as blocks, it gives an RBD design. So, latent scale design is the RBD design in rows as well as in columns. So, this is the layout plan of the latent scale design. So, in the first case there are 4 treatments. So, design will be 4 into 4 LSD latent scale design. So, there are 4 rows and 4 columns, and the 4 treatments have been allocated to all the rows as well as all the columns. So, in the second case there are 5 treatments. So, here the design will be 5 into 5 latent scale design. So, there will be 5 columns and 5 rows. So, in agriculture, if the suppose number of treatments are more, then this design is not applicable. Then analysis of variance table for the latent scale design. So, treatments are 5. So, design will be 5 into 5 LSD. Rows will be 5 and columns they will also be 5 as given in the layout. A, B, C, D in the rows, B, C, D, E, A, C, D, E, A, B, D, E, A, B, C, E, A, B, C, D. Similarly, if we see the columns, all the treatments have been allotted to the rows also, columns also. A, B, C, D, E, B, C, D, E, A, C, D, E, A, B, D, E, A, B, C, E, A, B, C, D. So, in case of the analysis of variance table, here source for the treatments, it can be calculated as V minus 1. So, we have 5 treatments. So, the error degree of freedom for the treatments will be 5 minus 1 is equal to 4. In case of rows, it will be also 5 minus 1, 4, because there are 5 rows. Columns are also 5. So, error degree of freedom will also be 5 minus 1 is equal to 4. So, here error can be calculated by multiplying V minus 1 into V minus 2. So, that is 4 into 3 is equal to 12. So, total error degree of freedom can be calculated as V square minus 1. So, if we square our number of treatments are 5. So, 5 square minus 1, 25 minus 1 is equal to 24. It is the strip plot design. It is also called as a split plot design. It is used when the 2 factors they require the large plot size. In case of split plot design, when only one factor require the large plot size. Here in this case, two factors they require the large plot size, both in case of tillage as well as in case of irrigation. So, the precision for measuring the interaction effect between two factors is higher than the split plot design, because the interaction block is the smallest in this case. Each block is divided into number of vertical and horizontal strips depending upon the level of the factors. Vertical strip plots for the first factor and the horizontal strip plots for the second factor. So, vertical and horizontal strips are always at perpendicular to each other. So, it is the layout plan of the strip plot design. In this case, we have treatments like tillage as well as the irrigation. In case of tillage, they require larger plot size. In case of irrigation, they also require larger plot size. So, then we will go for the strip plot design. So, tillage as is the main plot, it is in the vertical, horizontal is the treatment for 2, that is the horizontal. So, horizontal is the tillage and vertical is the irrigations. So, but we keep replication 1 and the 2, they can be like this away from each other. So, the ANOVA table of the strip plot design in case of treatments, A is the 5, treatment B is 5, 4 and the replications are 3. Here we have 3 errors degree of freedom. In case of split plot design, we have 2 error degree of freedom. So, error degree of freedom. So, in this case the replications error degree of freedom is r minus 1. So, replications are 3, it means 3 minus 1 is equal to 2. Treatment A minus 1. Treatments are 5, 5 minus 1, 4. Error A will be calculated as r minus 1 into A minus 1. 2 into 4 is equal to 8. Treatment B, the error degree of freedom will be B minus 1. Treatments are 4. So, 4 minus 1, the error degree of freedom for treatment B will be 3. So, then error B, error B will be calculated as r minus 1 into B minus 1, that is 2 into 3 is equal to 6. Then interaction A into B, A minus 1 into B minus 1. So, 4 into 3, 12 will be the error degree of freedom for the interaction. And error C, r minus 1 into A minus 1 into B minus 1. So, 2 into 3 into 4 is equal to 24. So, the total error degree of freedom can be calculated by multiplying r into treatment A into treatment B, and we have to subtract 1. So, it is 3 into 5 into 4 minus 1, 60 minus 1, 59 will be the total error degree of freedom. Thanks.